So what's the process for qualifying impurities? One is you need to take into account all data that's available. Toxicology studies, if they exist, what's the no adverse effect level, no AL. Also, a lot of these impurities are also metabolites. And so do you see these metabolites in animals? If so, it's qualified if there's good exposure to that metabolite. The other aspect is what are other products that are out there? What are other impurities that are out there? What's the information in the literature that I can glean, that I can use for helping qualify and ensure the safety of that impurity? Now, if no data is available, you're going to need additional studies likely to, or you're going to need to control that impurity at a level below where you need to qualify it. But a lot of times you can be very, very creative here, and especially important with regard to a very potent compound, especially one example would be a hormone-like compound, which may have a lot of different impurities. And if the toxicology is related to the activity, the pharmacology of the molecule, you can look at the impurities and assess whether those impurities have certain same type of potency or where the potency is relative to the drug substance. So if they have activity, you can also do qualifications to assess, well, there's not much activity there, it's structurally similar, you could qualify based on some of the in vitro potency. We actually did that with a product that was a hormone-based product and used that to help qualify the impurities. So be creative and science-based, but the studies that you have to conduct studies, they're outlined in the ICH guidance, and you can probably all have read these, but it's one species, usually the most sensitive. Obviously, the most sensitive species has to be tested with that impurity, or to get it or to have it qualified. Usually, a duration of 2 to 13 weeks. Very important to have a no adverse effect level. If you do doses of the impurity and you do see toxicity at all doses, you would have to do it again. You get a no adverse effect level to assess the safety margin of that particular impurity. Plasma exposures of the impurity are typically not measured. So you're dosing the animal with the impurity. Typically, you're basing your qualification, your extrapolation across species, on the dose given and not on the plasma exposure, which is a little bit different when you're looking at safety of a drug, of a particular drug substance. And this is perfectly okay to do this particular aspect of extrapolations. I'll get more into that, give some examples of that. The guidance, the Q3 guidance, also talks about having your impurity in genetic toxicology test. And if you looked at the new guidance, the M7 mutagenicity or genotoxic impurity guidance, they're kind of at odds with each other, really, and that should really trump anything that is generated from your earlier studies with following the Q3A. So I'll talk about this this difference, but basically your genotoxic assessment really comes out of M7 and not in vitro testing from Q3A. And I'll talk a little bit more about this particular aspect. And I'll give some examples of that. But you set your impurities up in the different classes, and if you do have a structural alert, it does not hurt you when you're in phase 1, phase 2. If you do have a structural alert, if you're beyond that, then you want to confirm that structural alert by doing an Ames test. Now, if your Ames test is positive, that's the definitive test that tells you whether you have a genotoxic impurity or not. This slide really gives some of the considerations that you need to have. One is what we talked about with regard to spiking in the drug substance or using the purified impurity. The other is the margin of safety you need. So, for instance, if you qualify an impurity at, let's say, 0.3%, and the specification is 0.3%, there's no safety margin there. And And so if the lowest adverse effect level is a very steep dose response where you do have an injury, you may want to have a safety margin with regard to your no adverse effect level versus your specification. And that really depends on the toxicity that's seen. Dose extrapolation from animals to humans are typically either per body weight or a body surface area. And this is a allometric scaling factor. And sometimes compounds toxicity scales on body weight, sometimes it scales on surface area. And this is a big factor. I'm going to give an example of how this can really affect safety margins. And then risk assessment for the intended population. For instance, if you're going in to cancer patients, well, perhaps it's not that important with regard to risk, with regard to having a impurity that may be genotoxic, for example, if you already have an electrophilic compound being dosed to humans. So an example of determining safe impurity levels is shown here. And the example I'm giving is where the clinical dose is 250 milligrams per day, and on a body weight basis, which would be 4.2, and on a body surface area would be 154. And then the qualification study that was run was a rat study where the no adverse effect level was 50 milligrams per kg per day, or 310 on a body surface area. So the 3 impurities were found. In the first situation, we had 0.04%. The toxicology study dose based on this percent, which would just be this multiplied by 50, is 0.02. I have it shown here on both a body weight basis as well as a surface area basis, which is the brackets here. And then the human safety qualification level is simply the rat impurity dose divided by your human API dose, which would give you the kind of going back, would give you the percent that would be qualified in the human study if given 250 milligrams per day. And so what you see here on a body weight basis, you're at about 0.5, and then the drug substance is also 0.5. But on a body surface area, you're 0.08. So quite a big difference, in fact, about 7 with regard to human safety qualification level. But the specification in this case is 0.5. Same example for B, very similar, where you have almost up to 0.1. The specification 0.3, but on body weight was around 0.2, and similar situation here as well. So whether you use body weight or body surface area has an impact in this particular case. Now this example was in about a mid-stage development, and so we did use body weight, but then again there's some allowance with regard to the specifications that were used. So here we're kind of really okay because we're at 0.5, which early on in development would be sufficient. But I want to use this as an example of the extrapolation. The factor used can have a big effect on the qualification level, and also it's important to rationalize which one you're using and why. I'm not going to talk residual solvents. Very good, the R5. It was mentioned about option 2, and option 2, you can, it's ridiculous how high you can get a qualification. But I think the agencies understand that, and so I really want to go back to the statement. They really need to reduce these to a practical minimum and reflect in their contemporary manufacturing standards. And I think they look at that. And the agency knows option 2 is yes. So it's just something to be cognizant of. But they do a very nice job on the limits. And the same here, because I want to get to the gene toxic impurities, which everybody seems to be interested in. And again, the elemental impurities draft, also very good, classifying different elemental impurities. And these are, again, this draft guide is very helpful and very practical, and I think helping a lot with regard to guidance and how to control.